Look closely at an unopened flower bud or the inside of a sliced tomato. The petals and seeds follow a recurring, strict geometry. Botanists use two specific blueprints to classify these patterns. On the left is Aestivation, which describes how sepals or petals are arranged inside a floral bud before it blooms. On the right is Placentation. This refers to the specific arrangement of ovules as they attach to the nutrient-rich placental tissue inside the ovary. Mastering the 5 variations of these two patterns is the key to identifying plant families and securing marks on board and NEET exams. We classify floral buds and looking at how the petal margins interact. In valvate aestivation, the margins meet exactly end-to-end without any overlapping. You will find this in Calotropis and the Solanaceae family. Twisted aestivation follows a regular pinwheel direction. One margin of a petal overlaps the next, while its other margin is overlapped by the petal preceding it. This one-in, one-out rule is seen in China rose and cotton. Imbricate aestivation features an irregular overlap. Out of 5 petals, 1 is completely external, 1 is completely internal, and the other 3 are partially overlapped. This is the standard for cassia and gulmohor. Quinquential aestivation is a more specialized subtype. Here, 2 petals are completely external, 2 are completely internal, and only 1 is mixed. You will see this when examining a guava flower. Finally, the pea family uses vexillary aestivation. It consists of 5 unequal petals, 1 large standard overlapping 2 lateral wings, which in turn overlap 2 fused petals that form a boat-shaped keel. Identifying these specific overlapping margins transforms a complex flower bud into a predictable, organized structure. Inside the ovary, placentation tracks where the seeds develop. In marginal placentation, the placenta forms a continuous ridge along the ventral suture, and the ovules grow in 2 distinct rows along that ridge, just like a pea pod. Axial placentation moves the attachment to a central axis. In this setup, dividing walls called septa split the ovary into multiple chambers. You can see these distinct chambers when you slice a lemon or a tomato. If those dividing septa are absent, it is free central placentation. The ovules remain on a central axis, but they occupy a single open chamber. This is characteristic of dianthus and primrose. Parietal placentation occurs when ovules attach to the peripheral outer wall. In plants like mustard, a false septum called a replum often forms, making a single chamber look like 2. Superficial placentation is a rare type where ovules develop all over the inner surface of the partition walls in a multi-chambered ovary. Water lilies use this to provide a massive surface area for attachment. Each of these 5 arrangements provides the structural framework a plant uses to house and develop its seeds. Let's test your eye for detail. Pause the video and look at these 2 unlabeled cross-sections. Which is free central and which is parietal? Focus strictly on the point of tissue attachment. On the left, the ovules are attached to the outer wall. That's parietal. On the right, they are attached to the center without dividing walls. That's free central. Looking past the empty space to identify the exact tissue attachment point is the most effective way to avoid visual traps on an exam. You will frequently encounter matching questions that pair these anatomical terms with specific plant examples. Use an anchor fact to save time. For example, knowing that a pea is both vexillary and marginal allows you to eliminate most incorrect multiple choice options immediately. Screenshot this cheat sheet for your final review. It reduces each complex definition down to the core 2 to 3 word rule you need to memorize. Mastering the rules of petal overlap and ovary architecture allows you to identify and classify plant families with anatomical precision.